Hello, my name is Grace Inamorado. It's August 7th, 2023, and I will be presenting on the anatomy, physiology, and communication of the retina. So taking a closer look at the retina is a sensory organ of the eye, and it contains the photoreceptors. Photoreceptors transduce light energy into electrical signals, which will then be converted to visual images via the neural pathways of the optic nerve. So if we take a look at the image of the eyeball over here, we can see where the image is zoomed in to show where the retina lies within the eyeball, and then it's further zoomed image of just the retina so that we can see which cells are involved over here. From the most external tissue to the most internal, the sclera right here is the outer connective tissue of the eyeball. The sclera is followed by the choroid layer, which contains the blood vessels that support the eyes. And then the retina is for visual processing right here. And then we have the vitreous chamber of the eye, which helps maintain the eyeball shape. The retina is composed of a pigmented epithelium. We're over here now at the more zoomed in image. Rods and cones, which are not labeled here, but they're there. They are the photoreceptors, and then we have the bipolar cells and then the ganglion cells, which converge to the optic nerve. Here is a nice large diagram of the organized layers of the retina. Again, we have the most external layer over here going internally. The pigmented epithelium here absorbs extra light and prevents light from reflecting back and distorting any images that we have. The blackish brown pigment of the epithelium actually comes from granules of melanin. Then next we have our photoreceptors, and here we can see that the cones are purple, rods are yellow. Cones are for color vision, rods are for monochromatic vision. And they are neurons that convert light energy into electrical energy. They release glutamate to begin the signaling process. So glutamate is important in the process of visual signaling processing. Excuse me. Signal processing is conducted via convergence, in which multiple neurons synapse into a single postsynaptic cell. So as you can see, you start off with more and then you end with a smaller number of cells. These are the cones and rods mentioned previously, and we'll discuss them further on the next slide. Then we have the horizontal cells over here, which synapse with the photoreceptors and bipolar cells to mediate lateral inhibition of the retina. And lateral inhibition enhances stimulus location and contrast so that an image can be seen clearly. The amacrine cells over here modulate information between bipolar cells and the ganglion cells. And these are the neurons where signals from rods and cones are integrated. So the amacrine are support cells. Glutamate released from photoreceptors onto the bipolar cells begins the signal processing, as we had said. And then the ganglion cells propagate visual stimulus to the brain via the optic nerve, and their axons all converge together to form that nerve. Now that we know the names and functions of the retina cells, we can talk about how light energy is converted to electrical signals. This happens during the process of phototransduction. As mentioned earlier, the light hits the pigmented epithelium, looking over here again, which is in contact, with the outer segment of the photoreceptors that we see here, which are the rods and the cones. Again, cone is purple, rods are yellow. Rods are activated in low light and they provide night vision, and they outnumber cones 20-1, which makes sense because if they're night vision, you would need more of them to be able to see clearly. At least that's how I see of it. The rods contain rhodopsin, which is composed of retinol, a vitamin A derivative, and opsin, which is a protein embedded in the membrane of the discs. And we can kind of see here, taking a look at the little diagram of the discs, we can see that the rhodopsin molecule is here with the retinal and the opsin. So going back to the rods, well, the retinal, is bound to opsin in the absence of light. But once a light photon is detected, the rod changes conformational shape and it releases itself from the opsin. This process is known as bleaching. Cones over here are for high acuity vision, color vision during daytime, and the fova, which was mentioned previously, has the highest density, which is mentioned for the first time, has the highest density of cones and the sharpest vision. And the cones also have 3 pigments closely related to rhodopsin, and they're excited by different wavelengths of light that allow us to see color. The cone colors are red, green, blue primary colors of visible light. One pathology to consider is color blindness, and this happens when a person inherits a defect in one or more of the 3 layers of the cones. So color blindness affects cones, C. I really enjoy this diagram because this helps me to visualize what I'm learning. So looking at the image over here, we can see that the melanin granules that were mentioned previously are located inside the pigment epithelium, and that's what gives the dark pigment of that epithelium. The pigmented epithelium comes in contact with the photoreceptors as seen here, the outer segments with the discs, as we mentioned previously. And purple photoreceptors cone, yellow's the rods. The outer segment of both photoreceptors are where light transduction takes place. So this is where light transduction takes place, phototransduction, And the inner segment is where the major organelles and metabolic operations of the photoreceptors take place. And then lastly, we have the synaptic terminal down here that sends the synapse to the bipolar cells to start signal processing. Let's take a deeper dive into the phototransduction in rods. Rods contain 3 major cation channels to conduct action potentials through the optical pathway. The cyclic nucleotide gated channels, or CNG, that allows sodium and calcium to enter the rod. We have potassium leach channels that allow an efflux of potassium, and then voltage-gated calcium channels that regulate exocytosis of neurotransmitters. So in the darkness, rhodopsin is inactive and cyclic GMP increases. The CNG and potassium channels are open, and there's a greater influx of sodium and calcium than potassium efflux, which keeps the rod depolarized around negative 40 millivolts. Looking at the image on the left, we can see that the G protein transducin, so it's this little guy over here, which we can see the transducin, and this is actually interesting because it's a G protein that is closely related to the G protein found in the type 2 taste receptor cells in the tongue. Just a little fun fact. Transducin is activated by bright light, and cyclic GMP decreases and closes the CNG channels. I also missed that the voltage gate channels over here are open and continuously release glutamate. So in light transduction, this occurs via the second messenger cascade, and this is when transducin is activated by bright light and cyclic GMP decreases and closes the CNG channels. Dimmer light causes a graded response in proportion to light intensity. Potassium continues to leave the cell, which hyperpolarizes the rod and decreases glutamate release. After activation, retinal diffuses out of the rod and is transported to pigment epithelium, where it reverts to its inactive form and it's recycled, so it's continually used. The recovery of rhodopsin from bleaching can take some time, and this is why there's a slow adaptation of the eyes from moving from bright light to the dark. Signal processing begins when the released glutamate from the photoreceptors contacts the bipolar cells of the retina. Bipolar cell signal processing is also modified by input from the horizontal and amacrine cells. On-bipolar cells are activated in the light when glutamate secretion by the photoreceptors decrease. On-bipolar cells are inhibited by glutamate in the dark and have metabotrophic glutamate receptors. They hyperpolarize the cell when the receptor binds to glutamate in the dark, and they are depolarized when inactive. Off-bipolar cells are excited by glutamate release in the dark, are inhibited in light with little glutamate release, and have ionotropic glutamate receptors that depolarize the cell by opening ion channels in the dark. Our next slide looks at the visual receptive fields of the ganglion cells. So next, the bipolar cells synapse with ganglion cells, and they receive information from a particular area in the retina known as the visual receptive fields. At the fova, which we remember has the highest number of cones and has the greatest visual acuity, the receptive field is small, and there are a few photoreceptors associated with each ganglion cell. At the edge of the retina, multiple photoreceptors converge into a single ganglion and result in a vision that is not too sharp. There are two ganglion receptive fields that are shown in the image to the left. There is the on-center, off-surround field that responds most strongly when light is bright in the center. If the bright light is off-center, the cell is inhibited. The off-center, on-surround field responds most strongly when light is bright in the surround, and if the bright is light in the center, the cell is inhibited. So it's the opposite of an on-cell, on-center, off-surround. An advantage of using contrast of light is that it allows a better detection of weak stimuli. So that's why we have the visual fields with the brightness of light. That contrast is what allows weak stimuli to be detected. And then we have the two predominant ganglion cell types. The M cells are large, magnocellular, and are sensitive to movement. P cells are small, parvocellular, and are sensitive to fine details such as texture of objects. Our last slide here goes over the processing behind the retina, and now that we have sent through the signal through the retina, the brain processes the information into visual images. Ganglion axons converge to create the optic nerves, and the optic nerves enter the brain at the optic chiasm, where fibers cross on either side of the brain for processing. So that's down here where they cross. Most axons project to the lateral geniculate body of the thalamus, which is here. The optic fibers synapse into neurons leading to the visual cortex in the occipital lobe in the back of the brain, in the back of the head, or brain. The lateral geniculate body is organized in layers that respond to different parts of the visual field. So we have a topographical organization, meaning information from the adjacent objects are processed together, and the visual cortex merges monocular information from the two eyes to give a binocular view of our surroundings. Our sense of depth perception depends on binocular vision. The binocular zone is the central portion of the visual field where left and right sides of the eye's visual field overlap. The two eyes have slightly different views of objects in the region, and the brain integrates the two views of a 3D representation. The monocular zone is where an object falls within the visual field of one eye, and these images are viewed as two-dimensional. I hope that you enjoyed my presentation. I enjoyed learning about the retina in great detail, and I thank you for your time.